charged, otherwise I will be charged. Okay. So when Professor Nagaraj said that we'll be having some kind of discussion on litigation related project, I thought I had to talk something about the work which we are doing. A, B, I thought Dr. Ramesh also will be talking about the work related to reading and all that. At the end I realized that Ramesh's name is missing and I have to talk about the story on virus which I don't work on. When I heard about this particular story, it becomes VIRUS for me. What is that? VIRUS? Vital Information Resources Under Siege. So I don't know what to talk about virus. But then I made a small story to talk about virus. Yes, so the story is this, that you are working on virus. Dr. Maruti said we are all working together to mitigate virus. So therefore the project is mitigation, right? Mitigation. So I feel there is some problem with our thinking. Excuse me, Dr. Maruti. This is how I think, all right? What I thought is we should work in PPP mode. So PPP mode with industry, that is happening here in this project. Public-private partnership. In this project there are more than 28 plus partners, institutes, industries, international partners working. But then what I thought is physiological plant pathology component is missing. So the PPP new word would be physiological plant pathology. And you should look at the angle of plant, not from the angle of virus, which we are looking at now. So that's what I thought. I'll just discuss. And again, I request all the senior faculty members to leave the hall because I might talk something nonsense. I have come with some normal physiology. I don't want to go out with abnormal physiology. You understand what I mean? I should not pick up some infection and go back. So this is about vertigation project which Dr. Maruti highlighted to us. What is that we have? We have some kind of, creating the stakeholders, interaction with them, and we have innovative diagnostic tool being evolved, and we have some kind of programs on plant virus vector interaction studies, and disease spread and climate influence on that, which Dr. Maruti did mention about temperature and the vectors population, and of course how to manage them and train the value and all kind of he said about extension related activity. So ultimately we have management strategy listed there. But a major component what I felt as a physiology is that I have to make this particular plant happy, and if it is happy, if it is protected, with respect to the defense what is there inside, probably we can manage the virus most effectively. Is the thinking of mine. Correct me if I'm wrong. Correct me if I'm wrong. Okay, that's right. So what is that we are talking? Why did plant virologists fail to control the viral disease in crops? Dr. Ellen Reddy is reporting virus every month. Right? Yes or no? His computer is full of virus genome. I understand that it is leaking, data is leaking all over the corridor. So what's happening? Why are you not able to manage? And virus like Ivanovsky and Pejavik discovered somewhere maybe 120, 130 years ago, but still you are playing with this 11 or 12 genes. Is that right, Elena? I don't know how many genes there are. But then why are you not able to manage them? And you pathologists are playing the virus like a cricket ball. Yes or no? Throw ball, whatever. You are playing with every virus and you are discovering new virus, and our CV is also improving thanks to virus. What I felt is we have to protect my plants. My physiology should be perfect. So you ignored physiology. So it's time for us to get connected. So that is the idea of mine. When we ask why are you not able to have you kind of resistance, people talk virus diversity, evolution, but that's happening. You know, coronavirus. Coronavirus also has a beautiful laboratory R&D center. Therefore, it evolved and produced new strains. So same in every virus, every biological system, there is some kind of improvement happening. So what I felt is, well, we are fighting war. When enemy is getting ready, we should also get ready. That's a plan we should work with. So that means you have to take lesson from different sources, try to organize our thinking and how best we can mitigate and mitigation can be more productive. Is a thinking of mine, Dr. Maruti. Correct me. with animals for billions of years during the course of evolution. They evolved multiple ways and made sure that run away from the situation or fight or fright. This is how it is. And this cartoon is very nice. There is no exhaust pipe there. There probably has solar system of running in the forest. So that cartoon also there is some kind of innovative thinking. So in other words, we have to learn from other systems. Animals avoid becoming thinner. So can animals can give us some lead ideas for us. It's a thinking of ours. So you have now similar kind of visual, chemical, auditory, tactile or vibration senses. So a lot of behavioral changes which you see to make sure that they are not getting trapped and become a dinner. I was giving a talk last month in some city college or city university. So I was introduced as a professor of crop psychology. And from there I got an idea. I used this like behavioral changes. I need changes psychology to be changed. Plant psychology to be changed. You virologists know only isolate virus and play with the mitigation strategy. So I thought I'd change the psychology so that we both can join together to make our plant more robust and tolerant. So I'm a professor of crop psychology also. So in other words, let's see, look at the plant, visual, chemical, auditory, tactile, even Mimosa has that kind of tactile element, right? So let's see whether you have any those kind of things. Maybe some of the plants which are outside will be talking or listen to my talk. The possible, that's how you should see, because they're all living organisms. Biological system, their language will not be, we cannot follow their language, but I'm sure there is some kind of communication happening. So that's what I thought we should see from the animal side, how animals avoid becoming dinner, how plants avoid becoming dinner. So let's see from that. Change animal with the plant. Let's see whether we can look into these kind of things in a different way. And you students, especially scholars, can be much aggressive. Ours is a sensing brain, it's calm. It's time for us to forget. Our plant stories are too many, we can only tell stories, but you have to think about innovative way of doing it. The plants provide nutrients for all your living organisms live within the plant body, which Dr. Marty mentioned in Neophytes, say they live in the apoplast, communicate with the plant cell and get some kind of benefits and transfer the benefits, which we call it as endophytes. So they give the space for the all kind of things to us also, food and nutrition, everything. And they have tough story because the way I showed the cartoon, their animal cannot run away, they have to manage everything within the system. So let's look at the plant, understand the problems, how these pathogen, bacteria, fungi, etc., when they enter, how they manage. Every living organism wants to complete their life cycle, make sure that genome is transferred to the next generation. So from that angle, I think we need to think aggressively. There are different ways of thinking, but youngsters can think and see what can be done to a plant and listen to its story, see whether we can manipulate certain processes which are very essential. Say for example, there are multiple layers of defense responses. You are studying already in your different courses. They are tightly regulated by multiple things. I'll just bias towards one story today, say for example, hormones. The hormones are called chemical messengers. They do communicate with these kind of environmental as well as endogenous pathogenic system, and they are involved in inducing the so-called immunity. They probably help us to avoid becoming inert with the pathogen or the things which we feel is not good to the plant growth and development. So, for the benefit of students here, I think plant growth regulators we call in general. I can put them in two categories, endogenous substances and synthetic substances. You can also play with them, but that's not the interest what I'm talking today. Endogenous substances are the hormones. They have specific biosynthetic pathways. They're produced in one part, probably transported to other part where they use specific physiological response. And one hormone is not doing the job. It's a combination of multiple hormones. We have identified only few. And you do have other substances which are also having similar biosynthetic pathway or the byproduct of certain biosynthetic pathway, and they are called the substances other than hormones. They also play a very important role in immunity, one difference what you are talking. In addition to that, we have similar kind of substances which are generated by industries called synthetic substances. They mimic hormones. They mimic hormones, they are not directly killing anything, but then they are mimicking hormones. They induce certain kind of processes which we feel is required. Say for example, there is virus infected leaf. You want to remove that. Probably I can spray certain kind of absorption inducing compounds and make sure that they manage. But then I should understand the process, which process and how. So you have endogenous substances, synthetic substances, put under the category of PGRs, plant excite, stimulate is the word which actually evolved, and all the students sitting here must be knowing about these type of hormones: auxin, gibberellin, cytokinin, subsystic acid, ethylene, rastenoside. List is growing, list is growing. We are adding one by one to the list of hormones. All the growth regulators are not hormones, but all hormones are growth regulators. The list is growing. If you go to the old textbook, only you see there's five, the big five, what we call. First discovered hormone is auxin. Then we started adding one by one, and all these hormones decide when to flower, when to drop. And that decision is indirectly influenced by the pathogens like virus and others. So now if you are able to talk to these hormones, probably they will help us manage. Even if there is a virus, probably we can manage the system in a more effective way. So they modulate growth and development. They are chemical substances. They are acting as a chemical messengers. As if they don't do anything, they just knock the door and go away. That's it. Then internal signals will activate and signaling process will start. So, in other words, single hormone is not doing the job. Dr. Nagaraj used to say that, sir, GA is more, ethylene is less. No. Don't think in isolation. That I teach in hormone biology course, one by one hormones. And while talking about auxin, I say that auxin is ultimate in grade, which is not true. Coordinated action is required. So you see that there is a coordination with respect to biosynthesis, there is a conjugation, inactive form, active form, it is metabolized and regulated in a multiple ways. So when there is a virus infection or a pathogen infection, you do see these kind of chemical so-called interacting factors, and which modulate the growth and development also, as well as they contribute for the disease tolerance or resistance or susceptibility. So in other words, coordinated action of these hormones govern. So we should see from this angle to make sure that my plant is not prey for the virus. So what are the different strategies you would have studied in your course? You have structural defense barriers, outer surface, maybe wax, resistance for the entry of pathogens or whatever. You have studied about the molecular mechanism, microbe-associated molecular patterns. Correct me, Dr. Elena Reddy and others, Padmaja is here. So in case if I say something wrong, because it's not my domain, you have bacterial proteins mediated some kind of microbe-associated molecular patterns. You have PTI, you know this already, pattern triggered or PMP triggered immunity responses. And again, you need a pattern recognition receptors. And you have several kind of the co-evolution strategies to make sure that you have ETS. To pathogens to continue their virus infection, they have to make sure that they evolve ETS models. You studied all these things in your course. You have strategies evolved with the plants with resistant genes or RF proteins. And ultimately what I want to say is that there's a continuous war going on between pathogen and the host plant. Both are living. It's called co-evolution. It's not that easy to manage, but with the help of endogenous chemical messengers which I've been talking about, there is a possibility that you can try to minimize the damage what you are going to get it. All these strategies are there, but we have an options. Say the SP Dinesh Kumar gave a talk, beautiful talk last couple of weeks ago in our department, and he did mention about some of these storylines that you have a receptor, you have something like PTR leading to PR gene expression, and ultimately sequence. And lot of things are happening within the cell. It's an ocean. The moment there is a pathogen, you have to see from a different angle. There are pathogens pushing some effectors, and again it is trying to push this out, and you have this pathway. Ultimately, some kind of defense or sensitive response is seen. That's what you've been talking about. But then remember, all these things seems to be coordinated with different types of hormones which communicate, coordinate, and sometimes when there is a pathogen, there could be some kind of signal amplification or development. As for example, in mitochondrial nutrient system, they again activate some kind of immune responses. Most of them seems to be linked to the hormone. That's what my understanding. What does it mean? It means hormone biology seems to be playing a very, very crucial role. And if you understand the biological process that are happening upon pathogen infection, be it the virus or any other pathogen, there is a way for us to handle the situations. Well, I was just wondering, there is so many things happening. When there is a pathogen, certain unwanted things are also happening. You have a big red show there in this cartoon, that is reactoption species. And these kind of things are not good for my plant. So plants shall also try to make sure that you suppress always some situation. Sometimes they have oxidative burst to make sure that some cells are committed to die out of aging. Death. Right? So these kind of things are again mediated by the important organelles like chloroplast and mitochondria. This ROS production is again assigned to particular organelles. They decide, oh, you decide to die. So this cell will die so that the virus spread is prevented. Who is deciding that internally? You would have heard about HR, right? Hypersensitive response kind of thing. So who is deciding which cell to die, how much cell to die out of aging-like situations? Again, there is a fine tune is happening inside when there is your pathogen is trying to knock the door. All kind of responses are activated. Sometimes you talk about systemic, moving from one place to another place through the flowing tissue and communicate. So in other words, the pathogen response is activated within the cellular system. You have diverse way of communication and also activation of systems like autophagy. Pathogen can also significantly influence the production. There is a co-evolution I mentioned. So what I actually found out from the literature when Dr. Nagaraj told me to mine and talk about this is that so you have virus or pathogens capable of hijacking hormone pathway or inducing hormonal imbalance. I told you already, hormonal imbalance are very important. So now these viruses are very, very smart, or pathogens are very smart. They try to manipulate these pathways. And they sometimes suppress the production of defense hormones. You got it what I'm talking? So I said plant have all kind of options, mainly hormone mediator option. I'm biased today. But then virus also this kind of additional thing, exploring and manipulating the host growth itself by overproducing certain type of hormones, maybe auxins or GM. You have options, transport, distribution. Most of the things are happening within the plant biology. Distribution, redistribution of hormones, conjugation, activation, inactivation, activation. Sometimes hormones are produced and kept in the cell, but they are getting activated as and when it's required. So these pathogens also interfere with that. That means they also have a good R&D team to find out which host is said that the virus is jumping from one host to another. That means that virus is a lot of R&D and make sure that that kind of pathway can be manipulated in that host, therefore I can go there also. So in other words, what I want to say that pathogen have a way of manipulating hormonal balance, imbalance, and changing even perception is everything. Hormones may be there, you can spray. Well, I say that auxin is good or ABA is good to suppress this particular disease, you can spray that. Then, if that virus or a pathogen is repressing the expression of a receptor, your application is based. Reception is everything. Receptors are very important to sense the signal because hormone is not a chemical, it's a chemical messenger. Receptor should see the hormone, then only action will start downstream. You see the change in the hormone receptor expression also there are reports. Although some of these reports are to be validated again, but then I started mining since yesterday morning. Nagaraj tortured me since yesterday. He didn't allow me to sleep last night. Okay, if I yawn in between, excuse me. It is not because of me, it is because of virus what he injected on me. So, this is what I want to think. Youngsters and students and new generation researchers must think about plant while you are thinking about virus. I just want to quote this example. So, for example, you have salicylic acid, everybody knows about this, right? So, there is a pathway. I'm sorry, those sitting in the last bench may not see this. You have chorismate to convert it to some X compound. And you have a compound, this X compound is not useful. This particular infection converts chorismate to some X compound so that this salicylic acid pathway is suppressed. Don't worry about a statement what I make, but what I have to say that there are certain effectors that convert chorismate into this and therefore it is not available for your SA biosynthesis. Don't you think it's very interesting? That pathogen, virus or XYZ did think about the biosynthetic pathway and attack that particular biosynthetic pathway. How much money it would have taken to do the research, we do not know, but it is successful. So what I want to say is that not only that, there are several pathogens, they have their own SA biosynthetic pathway actually something else comes so that they are okay to get ready to fight. So you have essay production antagonistic way of doing things. So you simulate, mimic, and make sure that you bypass the defense pathway and get into your infection. So these organisms are very smart, really. And I have another example. You know that ACC oxidase, methionine to right away to ethylene. There are different stages of ethylene production. Let's not talk about it. You have now ACC 1 amino cyclopropane carboxylic acid. ACC is converted to ethylene. Last stage, very important, later steps in ethylene production. So now you have certain things, they actually produce ACC, upregulation of ACC oxidase. That means I have ethylene getting produced. Ethylene is also required, it's a gaseous hormone required for several signaling and response as well as susceptible phenotype depending upon the situations. So hormone pathway seems to be being hijacked. Perception is manipulated. You got what I want to say? So these kind of innovations are very important for us to manage even in the programs like litigation to manage the viruses. Auxin, say for example, some of you are aware of this. You would have taken. So you have auxin and there are certain kind of repressors, they have to be degraded if you want to get the auxin response. When there is no auxin, repressor is bound to this kind of auxin response, cis elements and all that, they don't allow. So the moment there is an auxin, it is guided for ubiquitinate mediated degradation pathways. Now you have certain organisms, they make more susceptible when there is auxin. So that means they are playing with these repressors. Auxin renders more susceptible for the plant, so that means they produce more auxin or induce the response of auxins. And people demonstrated that exogenous application of auxin leading to increased virulence or whatever. It's not just suppression of production, it could be enhancement of the hormone. So it's not just auxin, I just want to tell you. When you change the auxin, I change the ratio of auxin to cytokine. I change the ratio of auxin to gibberellic acid. I change the auxin ratio to strigolectin. You understand? Racinosteroid, these different hormones. So that's what it matters. We are biased, we are thinking on one glance, but there is a more complex events happening inside. So the same way repressor, I'm not going to talk, details about it, but what I want to say that there are certain kind of synergetic role with the so-called stress response or biotic stress response hormones like jasminic acid or salicylic acid with auxins. So they try to manipulate the core hormone pathways so that they can hijack and get inside and try to establish the infection. You heard about this. Pathology first course 101 itself you talk about this. Right, you have a Buccaneer disease through the seedlings in rise, tall. It's because of what? Some fungus. What it does? Jugular Physiocori. I think the name has been changed now. What is the current name? Jugular Physiocori name has been changed. Yesterday I read, I forgot today. Anyway, it doesn't matter. So what I want to say is that jugular physiocori, what does it do? You have an elongated seedling, internodal length is elongated, great. Why? This fungus produces tons of GA. That's the GSOs what we get for spraying grapes, all kind of treatment now. GA is harvested from those fungus. Forget it. So why it is producing GA? Its role is not to elongate the plant. No. Can you think? That's how innovative thinking. I did talk to those fungus the other day. It only told me that. It is not producing GA for elongating the stem. It is producing GA for some other purpose. What would be the reason? It's written there. It wants to suppress GA activation. Are you getting the logic there? I don't know, you are not getting charged. What's happening? Are you not getting what I am telling? It's not producing GA for the sake of producing GA. It wants to block J activation. What is J? Are you getting the logic now? So this is how you should think. So that's fine. It is tall, elongated. In fact, if it is tall, elongated, plant will die faster. I don't want that. But then it is trying to to establish. So there are now these kind of thinking and papers are being generated, knowledge is being generated to ask the question and how to block. What is this DELA? DELA is a repressor protein. I am not going to talk about DLLA, which is one of the things which has to be erased if you want GA response. Okay? So it should be removed if you want GA response. So from that context, I just want to tell you that these pathogens and fungus or whatever I'm talking about attack hormones, chemical messengers. I think that's a major way of attack, so that they can gain entry and do whatever they want to do. While discovering Erinnerity Garu, while discovering virus, try to see this also.
We have required to come up with a manuscript replicating Plasmodium cycloride.
Very good, thank you so much. So I'm not wrong there. My thinking is correct. So I just want everybody to see from this lens. Don't think about only pathology, think about physiology. Got it? In this case, hormonal biology. So in other words, most of the so-called hormone-linked stories are in and around the major hormones like auxins, cytokines, or gibberellic acid. But then lot of information generated with respect to DELA, and they say that the DELA, integrates multiple signals. DELA means DELLA, it is a repressor protein, integrates multiple signals. Integrate multiple signals, and then ultimately you have G responsible, so ultimately you have some kind of tolerance mechanism, or if you have mechanism to work on those responses, you get a susceptible so-called phenotype. So, if you want to play with immunity, you have to play with hormone. That's the message I want to advocate. You agree with me? You got some lesson now? So, stress out of hormones is not my title. You want to read more about this? There are several review articles and papers popping up now. So, they are looking at physiologists. Thank you very much for discovering new problems. So, we have a solution. So, you can probably read this article, or you have some reviews available. You might get better ideas than what I do, because you work on pathogens. So today, I just thought I'll take a little bit, deviation, then I came back to the virus. Those pathogens, what they played with, maybe bacteria, fungus and all that, relatively okay, you can manage, because there are two biological systems, you have some way of catching them. But this virus, I think Dr. Maruti also can educate me, difficult to catch. Yes or no? That is 11 genes, still they are there. 100 plus years, you are not able to do anything. They somehow depend on post-plant physiological process directly. Yes or no? Right? So you and I have a more responsibility. It's time for us to get married. With money, of course. No passive marriage, active marriage. So this is what I would say. So you have now viruses. They exploit the system. We do not know how they attack, the way they are attacking. So in other words, I should think about plant health. In other words, I should think about basic physiological processes, including photosynthesis. Right? So probably they attack this kind of basic process and try to do something. So it's our time, it's time for us to think aggressively on physiological basis of virus susceptibility or tolerance, which we are not. Alright? So this is what I thought I would talk and discuss with Nagaraju, and say, I was just searching here and there, I'm not getting good so-called validated data sets, although there are good information here and there. So one thing what I found is this CMV, cucumber mosaic virus. So you have this computer simulated models and all that, and this fellow can in fact infect more than 1,200 plant species. Is that right? CMV goes everywhere. So I don't know how he is entering and doing the job. He or she, I don't know. And most of the reports, they say that it is trying to something interfere with my basic process, that is photosynthesis. So you see a chloroplast diamond there, right? And it also has some kind of attacking the hormonal signaling. That's what some literature here and there. And some hormone signaling pathways are relatively well studied, like tobacco mosaic virus, which Dinesh Kumar was talking the other day. So you have now some basic physiological process as well as hormonal link being shown with these kind of viruses. So that's what I thought, this is a way for us to think about it. No, I was just wondering and trying to mine the information, how it is attacking the photosynthesis. So you have studied 101 course CPH, some of you, yes or no? Right? So youngsters, I'm sure, crop physiology or plant physiology course you would have studied if you are from Horticulture University sitting here, and they would have talked about one protein called FD, electron transport chain paradox. Hey, Elena Reddy, you should keep quiet.
One more paper is there, sir, but we didn't prove the cancer biological validation of the
You
experiment.
didn't get married to me, otherwise you want to be isolated, so you will isolate.
Yeah, I'm sure that in
The problem with virologists is that they have identity crisis. If you solve the problem, then you may not have problems. Anyway, that's good. I'm very happy. My thinking is on, right? Huh? Ramesh Garu, boliye. Anyway, what I want to say is that paradoxin is one of the important proteins. And as a professor of physiology, I will be very happy to talk more. I not. So it is electron transport chain internal protein, whatever it is. So this particular protein is produced. You have something called messenger RNA, then you have transit peptide, whatever it is, processed and guided to the chloroplast, and it is assembled. And it is a component of electron transport chain. So I have PS2, don't worry about all that, photosystem and blah blah blah. It is guiding electron transport ultimately. Very, very important protein. If something happens to it, your electron transport, photosynthesis called light reaction or photochemical reaction is disturbed. And it can, if the protein is not properly formed, misformed, you have misguided electron generated. So electrons are actually guided in a proper way and random. And ultimately you know what's happening. Let's not go to the photosynthesis here. So I have an option. If you have misguided electrons to generate react oxygen species, then you know what happens to react oxygen species. If it is oxidative burst, if I don't have defense machinery like SOD, catalase, peroxidase, blah 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 getting activated simultaneously, it's kind of suicide. Now what they have shown is that your protein, what is CAV? Okay. What is CP? Students should tell me. Gold protein. You see what gold protein is doing here? Right. It is actually targeting, I don't know which research paper it read, that FD is important. It is attacking right there and make sure that you do not have FD protein, right? This leads to increased H4 level, blah blah blah. You have intervenous fluorosis of bleaching, photo bleaching, etc. etc. Don't you think it's very interesting? So understand the basic physiological process and look into the virus. What is it interested in? There are many people here working on docking, right? Molecular docking. Data is there. Probably it's time for us to see from that lens. Look at different proteins like FD, 3D structure of FD, and see what is happening with the CMB, some blah 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 CP or whatever it is. See how they talk to each other, whether you can actually stimulate and generate some novel information. See whether these pathways are operating in different ways. I can tell you more, but you have to pay for me. I'm a businessman now.
Now that is published, sir.
Huh?
Now that is published, they have shown that.
You have shown. Very good. Excellent. So I didn't copy from you. So, what I want to tell you is that there are different way of looking into it, and look at the plant biology, plant physiology. Then look at the virus also simultaneously. Don't work in isolation. And these kind of interactions might help us to address the issues of mitigation. Okay? So you don't have to mitigate, sir. It will probably talk it. Alright? So, are you getting the logic now? How much time do I have, sir? 10 minutes. Is it okay? It's boring now. So, in virus also I said plant health related, all these stories are also there, and I request those who are interested to work on these issues. You have ethylene, jasmonic acid, salicylic acid, they seem to be playing crucial role. Look at them, oxin, abscisic acid, cytokine, gibberellin, where every hormone is there. Don't think in isolation. inflammatory hormone, hormonal imbalance, is actually the culprit for susceptibility of this virus. So I was just reading through some of these reviews, and wherever I could understand, I understand that Brasinus, right, to salicylic acid, to cytokinins, all of them are in one way or another way linked. And most common thing seems to be SA signaling with respect to virus, what I could understand. Similar to SA, cytokinin, PR also, PR means Brasinus steroid-based hormone, recently discovered and characterized. We have good signaling pathways identified with respect to PR, Brasinus steroid, and that seems to be very, very important with respect to virus infection as well as tolerance mechanisms. So you have some stories with respect to auxins. If you spray auxins, they become susceptible. Same thing being reported with respect to virus also. So, and there are certain mechanisms by which these virus also attack on your auxin distribution. There are proteins called pink proteins, pink-bound proteins, which are involved in auxin transport. And some of these TME, tuberculosis virus, disrupt distribution, and therefore it gains entry with respect to susceptibility or sensitivity. Well, so the virus seems to be attacking phytohormone in different way, and SCA, say for example, salicylic acid, whatever the literature information what I have is probably playing with the replication related storylines. And hormones like abscisic acid is playing with endogenous beta-1,3 glucanase inhibition through which you have these callose, whatever it is, they are going to be degraded for plasmodesmatal connections and movement from one systemic movement from one side to another. That seems to be disrupted. And you have therefore ABA playing some kind of important role with respect to virus resistance. So I feel that there are a lot of information which can be mined and used as a strategy to manage virus. So in other words, you have a positive role as well as negative role of these hormones. Please make sure that we see in a holistic way, not in isolation. Isolation may not give anything. All hormones are important. No hormone is bad, but then their ratio, relative abundance, distribution, redistribution matters upon virus infection. Systemic movement of virus depends upon the distribution, redistribution. One thing what I found, Dr. Maruti, is that there are conditional mutualism with respect to virus. That's interesting. We talk about these days combined stress tolerance. Say for example, drought. Virus mediated drought tolerance. That's interesting. Yes or no? Right? In fact, TME infection increases ABA level. ABA level can actually make, well, water conservation strategy, I think you are aware of it, stomata get closed, right? ABA also has some kind of mechanism of minimizing transpiration by triggering the production of waxes and deposition of waxes. You have lipid transfer proteins transported and deposited on the leaf surface. So you have more hydrophobicity, cuticular transpiration is reduced, ABA closes stomata, stomatal transpiration is reduced, right? So ABA also induces desiccation tolerance through LEA, like late embryogenesis, like abundant, like desiccation responsive protein. So overall, you have mild virus infection can also create these kind of benefits. So don't think that virus is bad, it's also good. Depends upon the situation. So these kind of stories are also, I say that conditional mutualism has been reported, and that's also a very important story for us as a physiologist to take further, because we are interested in managing the combined stress in our situations. So I found this kind of storylines, and we also work on endophytes. We work on fungal endophytes with Professor Nagaraju and others, see whether they can improve overall plant health. Therefore, it will not pick up infection. So here is a story which I picked up from the literature, is that you have virus mediated drought tolerance, and study is limited here. I have endophyte fungus. So when you have only virus, you can have some kind of stabilized photosynthesis and conservation of water. RWC means relative water content, and it produces compatible osmolalite to make sure that cell is maintaining its cell level tolerance upon stress induction. And you have fungus plus the virus, similar thing. In other words, it is somehow endophyte is making virus to be comfortable, and you see only no fungus, no virus in that cartoon, so it's a sensitive phenotype. So these type of information are also being generated, but the information, the data set is very limited, but then there are ways by which we can look at this angle also. Is it okay? Agreed? So, I say that fungal endophytes and endophyte mediated, I'm not talking about any data which we have now, maybe Nagaraj or others might discuss about the endophyte and mediated so-called virus resistance, but there are ways by which we can do, we do have something. So in other words, you have to look into the literature, you have so many information being generated, so you can use the virus, plant pathogenic fungi, a promising biocontrol agent like situations. I have pathogenic fungus converting into a beneficial fungus with the help of mycovirus. That's interesting. I'm not saying this is a way, this is a good strategy for mitigation, but whatever I talked earlier is to make sure that you improve the immunity and resistance within the host model system. So endophytes is important. And now that I am talking about this everywhere is that if I have a good plant physiology, I will have a good human physiology also. If I have a good plant physiology, meaning what? My plant is doing good with respect to growth and development, you have a quality fruits. So that quality fruits as well as good endophytes, which probably we eat through salad, also contribute for our endogenous microbiome. This is not my data, it is everybody's now being promoted. So the system is totally connected, it's not disconnected. So if you have a good plant physiology, you eat good food. That means your biological system is normal. You won't fight with your friends. You can have a good night's sleep. You understand? If you want to have a good night's sleep, make sure that your plant physiology is normal. If you do not have one, then you have all kind of problems which is highlighted there. So if you have a good plant physiology, you will also have less probability of infection. So ultimately you may not have to spend a lot of time for mitigation strategies. Please remember, all these things are connected. Please, plant health, human health, everything is connected. That's what we look at it. So in other words, we have to think in a holistic way, we have to join hands to make sure that we understand the complex network, then address the problem what we are on hand. So I just want to talk to you and impress you that we need to have a combi approach. Combined approach is important. Dr. Leonardi is generating based data. Let's see whether I can get connected with him to have a combi approach. And someone else, Professor Nagaraj is also looking for seaweeds and some kind of endophytes which we are. Let's see whether we have a combined combi approach. So crop is a biology. You see that cartoon what I put there. So many cyclic events are happening. Everywhere there is a communication traffic jam. Combining techniques of plant physiology to plant biology to virus, microbiology, everything is required to create a kind of new thinking and dimensions. So we, in vertigation team also, should think from that angle I feel. It's not one way of mitigation, we should also look into basic science. So another speaker in the last talk, he said, basic science is like Brahma. There is no Brahma temple now. So basic science is totally ignored. So if we do not have basic science, you have Lakshmi Saraswati temple everywhere, but not Brahma temple. That's what he was explaining in his talk. So we need to spend a lot of time on basic understanding so that properly can receive and don't work in isolation. And in isolation work we may not be anything. So therefore I felt this is very important. There is one big university, although there are many other places, but I like this. You see that the Department of Plant Pathology and Crop Physiology, interesting, right? If you want to attack pathogens, this is the way you should think about it. It is important. In fact, that university started, Louisiana State University, Dr. Nagaraj said, in 1924, they are working together. They have some interesting story to say, communicate. So in other words, this is how you should connect and join together and make sure that you understand the biology, then attack the problem what you have in your hand. So I just want to thank the entire vertigation team for giving us an options as a knowledge partners. So we should have been very happy with money, but unfortunately I know the difficulties. Professor Nagaraj managed the show, we didn't do anything. I and Ramesh is a passive worker. He used to go for lunch whenever he calls. Without payment, he used to pay for us. And I'm very happy that this PPT, Plant Physiology team, helped all these things without any funding. A lot of data is generated, and you gave me a chance to present my PPT here. Thank you so much.
Doctor, can I have few builds?
Sure.
Yeah, so myself Naveen, so I'm coming from United Genetics. So after hearing Dr. Maruthi's presentation and after your presentation, so what Maruthi mentioned, definitely, I mean, it still holds good, but your presentation really closed the loop now. It made a complete session today. The reason is actually because the way the dynamics of cultivation itself is changing globally, including in India. So more particularly in India, because of the huge diversity we have within India, this kind of technology is required. So basically, actually, till I'm also a pathologist, alumni of this department, and till 2016, I was
Welcome back to
also
the department. Yeah.
because I also started my career as a pathologist, and till 2016, I was thinking that breeding for resistance itself is the way to mitigation of plant pathogens. But in 2016, I was actually offered a role of heading biologicals for Asia Pacific when I was working in Syngenta. So that really made the complete shift in my paradigm of thinking or the managing the plant diseases, of course. So because the new technology which is coming is because if you think from a genetic perspective, biotechnology perspective, we are at the level of gene transgenic, gene editing. Of course, I mean, this technology is also required, but we also have to see the cost on the yield. So if you pile up more genes, of course, it will be at the cost of yields. Like the best example is the tomato.
Yeah.
So when we look into the variability which is present in begomoviruses and also the staking of genes, we have to TY6 now. But if I pull all the 6 genes in tomato, then it will be resistant to virus, but also resistant to yield as well. But when I think from the grower's perspective, when I think like a grower, of course, they want resistance to diseases, but also they want it a complete package. Virus resistance, of course, it is required, but that only doesn't serve the purpose of the grower. Yeah, because the entire industry we are running, it is a trait-driven research, basically. If I think from the grower perspective, actually we work for the grower traits, then the consumer traits, then traders traits, and now actually the new dimension is the nursery traits. So if I want to give a variety of the hybrid to the grower, they want a complete package of it. So unless actually we cover this area, understanding the physiology part. So if you look at from the industrial perspective, so to address this, a couple of things Maruthi already mentioned, like he mentioned the chemicals. Of course, in the current situation, we still cannot ignore chemicals.
Yes.
Though the European Union actually, they have already launched a drive of field to fork. So to complement actually that drive, the new technology like the biologicals also have been added. So under this biological umbrella, so biopesticide Maruthi mentioned, the plant extracts Maruthi mentions. Besides this, you touch the plant growth regulators. We call it as a PGR. That is also comes under umbrella of biologicals.
Yeah.
And besides this, the most important is the biostimulants.
Yes.
So because having worked on that for more than 4 or 5 years, so the next 20 years will be the era of biologicals. So that is going to come. So that is mainly the biostimulants. We have the amino acid extract products that are coming in the industry that regulates the physiology of the plant, thus indirectly help to build the immune system within the plant. So that will complement with the chemicals as well as the genetics.
Yes.
So thanks for presenting. This really was a very good session.
Thank you, sir, for adding on. And he put on one point which I didn't take, the storyline, nutrition. Nutrient management also plays a very important role in maintaining basic physiological process that make the plant to resistant or susceptible. That part I didn't mind. I have some story on that, maybe. So we cannot have everything, but then beautifully mentioned that this approach of biologicals as well as the improving goal of resistance by having good plant physiology is very important. Thank you, sir. So, any queries from students?
I have a question.
Yeah.
I'm Dr. Suvairita from MS Ramaiah. So I worked on similar aspects at my PhD level while I was doing an IAHR. So I worked on salicylic acid for 3 different viruses, CMV, CVME and leaf curl. So I got pretty good results, and one more thing which I observed during the review of literature is There's a lot of work done regarding this. But it is all restricted to the research lab and it is not coming to the field. So when it comes to the publication also, the virologist doesn't accept it. So how do we go about this?
Yeah. So I don't know, thank you very much. I don't know how to address that question. Can anyone help me? Students, others. See, basic strategic research are understanding the system, and that might give you durable resistance later. That's what I want to say. You may not have directly to industry application tomorrow, but then without which you can't move. That's very, very important. Basic strategic research. Certain strategy, you know what we are. Certain basic research, we don't know what we are getting, but we have to do that. There is no other route. Starting from invention of wheels to a light, electricity, everything, application comes later. You know, when did Raman discover Raman spectroscopy? Students can help me, do you think?
Actually, there was a questionnaire, something regarding the surveillance questionnaire and the story questionnaire and data analytical questionnaire. So one is to fulfill all these questionnaires, we can understand what is the cause and what is the mechanism.
You're talking the same line, I'll come back to you, okay? This is a different question, I think, no? Okay. So Raman discovered light scattering, right? And he won Nobel Prize, all that physicists liked it. But do you know the application now? Starting from our laboratory to the hospitals, everywhere it's application. So that was the basic science what it did, basic discovery. So application started coming much later. So we cannot ignore just like that. I know university mandate is to MK, networking and knowledge generation. That's all. Our goal is only that, networking and knowledge generation. Sharing, knowledge sharing. Networking is required to share the knowledge. From that angle, I think basic strategic research are very important. Their applications might come later. We cannot stop it. I don't know whether I addressed your query. Dr. Maruti, you have to add anything? That's what she meant. I don't know.
No, I fully accept what you were saying. I think maybe the problem with paper not being
Not just the paper, forget mine, but I think there's from 10 to 15 years back only this work is being done. Let's tell the induced systemic acquired resistance and things like that, and not just the phytohormones. There's a lot being studied, like the biostimulants and things like that. But what I observed is the application, as in the field, is also not happening. So when we talk, people go about the breeding techniques and the latest CRISPR-Cas and things like that. So this part, as so beautifully mentioned, is being least ignored actually. So my question is, how do we actually bring it to the field?
Yeah. So that's the interesting part, and
It's
it's good R&D.
attempts were made actually to synthesize synthetic canalizers like messengers like a salicylic acid and synthetic canalizers like isonicotinic acid and other things. But the problem is, I think, I can say when you spray translocation of this from so the
Moreover, you're right, translocation, distribution, A, B, overall biology itself
Yeah.
matters a lot.
So, and moreover, it's a complicated thing. For example, if you say QOLC, for example, I think this white rice saliva actually, so it will go into down-regulate the jasmonic acid and triggers. So you may be manipulating the plant to, can say, synthesize more jasmonic acid, but there is a signal molecules in white rice saliva which can down-regulate the jasmonic acid, which acts as a defense against, you can say, insects. So it's not that in natural conditions, it's more dynamic than what we assume. That's why sometimes all the things cannot be translated. Whatever the basic science outcomes cannot be translated into application. And as you mentioned Ramana, but a similar thing like you can say Higgins boson, here it was proposed, they were a law person in the 1920s. Now, yeah, so everyone revisiting that and going behind a God particles, and a big institute has been to study that the particles which move faster than light. The CERN is the institute which has been established. So sometimes today's nonsense, maybe tomorrow's more relevant stuff. So science, nothing, any information is actually a result, or it is positive or negative today, it doesn't matter. Tomorrow it may be having an application. So when need to wait and see. So,
Anyway,
it's
that's a
something
good point.
like you can say a
I
correct.
also appreciate we need to look at translation option also, because biology is more complex than what you think, so that will be way out, I hope so.
Yes.
She was asking, then I'll come to you. You wanted to say something, no? Can you? Questionnaire, why it is not translated, okay,
For any outbreak, it's a challenging.
alright, okay, thank you. Krishna, Shankarappa, sorry.
See, in a virus infection, especially vector-borne, we have interacting vector, endosymbiote, then a plant, a virus. How at a time we can, what are the strategies, thinking of one plant, one pathogen we can deal. You are in vector, it have endosymbiote. It is also play important role for transmitting or something else.
Yes.
Then vector also play important role physiology of the plant, and virus also play important role physiology plant. Plant also will have some role. Then and in this aspect, what we have to, what strategy with respect to physiology?
Good question, so we have to have a joint family.
Yes.
Entomologist, microbiologist, maybe zoologist if you require, right? So that's very good question, and that's how it is. The system is connected. It is not isolated.
So you only can include a physics for a detection for
Precisely. I mean, that's how it is. I want to tell you this way, the multidisciplinary, interdisciplinary way of doing things is the best way to do it. I just mentioned this before I conclude. I work with aerospace engineer. Can you believe this? Aerospace engineer. I didn't go to aerospace. So they were working on shock waves. I wanted to use shock wave to transform a bacteria by cloning something. We use electroporator to create shock wave to push the naked DNA inside. So I discussed with Professor Jagdish from Indian Cell Science. We did a lot of thinking, and they have a shock wave generator. It's a very big underwater current shock wave generator. And I use naked plasmid DNA, and E. coli cell DH5 alpha, push the naked plasmid DNA much efficient with transformation efficiency compared to electroporator. What does it mean? It means you should be open to talk to anybody, and biology is like that. And of course, that knowledge grew and did that. Jagdish, who was here last couple of days ago for a workshop, mentioned about these things. So let's not work in isolation. Let's join together, and I'm sure we can address the problem of virus or any other pathogen. Vertigation is one of the ways by which that is mitigation. So we have the other way of looking it up. So with this, I think it's 1 o'clock. I would like to thank Padmaja and Professor Nagaraju for giving me a wonderful opportunity to talk to you all. I'm happy that no one left the lab, hall. So you are glued to the chair. Thank you so much.
Thank you so much, sir, for such a wonderful presentation. Now I'd like to request the head of the department, and I also request Maruti sir to join in handing over a small token of love. Please, sir, please accept a small token of love.
So many of us are litigation members. We are exchanging. So I'm very disappointed because I had two videos. It didn't work here. That video would have given